Welcome everybody. My name is Jan Vastbinder. I'm one of the co-organizers of this unique event that we have in the coming 4 days. I don't have to introduce Professor Brenner, Sydney Brenner. You all know him, so I won't introduce him. I'll just give you a few words as to the procedures that we go through. We will have 1.5 hours today with Sydney. He may have a little break in between, but
there's some more people have come.
Yeah, well, they just fill in. That's okay.
Is this on?
So it's 1.5 hours, which is about an hour is talk, and then there's discussion after that. You'll all see how it works. Tomorrow we will do the same thing, except it won't be here. It will be on the 4th floor, mezzanine, in a different room, but you'll see the signs. And on Monday and Tuesday we'll have Terry signals. Come here. There's this place. There's this space here. The whole ceremony, the whole activity is in hands of Sydney himself. So he will talk, and I guess you want to. Can we interrupt it for questions, or not?
At their own risk.
All right. Okay. Well, I think that is a good start now to give it over to you, and
okay.
I hope you enjoy yourself.
Well, I thought that many of you should hear these series of talks, because on Monday, Terry Janowski will come, coming from La Jolla, and he will talk together with me. Because, you see, this is an accounting of what happened, the whole revolution in biological thought, together with its still the most important new problem, which is how does the brain work? And you should realize that the people who created molecular biology were also the people who went on to do things with the brain. Of course, they weren't alone. On the other hand, Cambridge, which is where most of this happened, was also a great center for all kinds of science. And so it's important to realize a lot of people's interactions, which were not formal in any way, but led to this being one of the significant developments and a big breakthrough. So I give this to you, I talk to you about the early days first, and tomorrow we'll talk about what one's concept of how to solve brain problems was. Now, there were two founders, of course, in the whole of the computational sciences, only one of whom was in Cambridge. And that was Alan Turing. Some of you who've done computation have heard of Turing machines, almost certainly. Turing was a PhD student and young fellow in my college, King's College, in Cambridge during the 30s. And he was interested in the subject, and of course you have to remember that people were building mechanical computers. There's another man who is tremendously important, and that's John von Neumann. John von Neumann is, in fact, thought to be the father of computing. But he was a mathematician at Princeton in the Institute of Advanced Studies. And in fact, that is where Turing went from Cambridge. He went to America, and that's the thing. However, there had been a rather long discussion with all of these things, and as you know, one of the power ideas came because one of the important things which we add to the fundamental sort of physical principles, which was matter and energy, which is what everybody was concerned about. How did you make the chemicals that were in biological systems, and how do you achieve, the energy? How do you keep away from thermodynamic equilibrium and death? And the missing element, as it turned out, was a new concept, entirely novel. It's a concept of information, which of course linked it with the computing thing. And the person who really pioneered that was first Turing and of course von Neumann. Now, it was, I think, von Neumann wrote a very important paper, which you should read. It's the theory of self-reproducing machines. Okay, what did you need theoretically so that a machine could reproduce itself? And that was written before the DNA, long before the DNA. It's a very interesting paper. It existed in notes that were printed before the war. It existed in notes, but it was finally published, and I have read the published version, which is in a wonderful little book, which you should notice, and which I'll put my copy in the library here in a few months' time. Now, what did von Neumann say? He had tried to think of how machines could actually build themselves. And he realized that you had to get parts and so on. But effectively, he didn't know. He had the idea there had to be a program, like a Turing program, in which one event followed on the other. And so he writes this, and in it, it's very interesting to read it. He says, like Turing had a tape that fed a Turing machine, he also has a tape. On this tape is written the sequence of events a la Turing that you have to accomplish to achieve the computation. But what he realized is you have to build the componentry of the machine, and the machine can't run unless it's got a tape, right, to program it. So he had added two more elements. One was a copying machine that copies the tape. And so after it's built the machine under the instructions of the tape, the machine builds itself. It then makes a copy of the tape and inserts it in the new machine. All right? And that business that you have to transfer the information as written on the tape into the new machine is absolutely fundamental biology. You don't do it only by self-reproduction, but in this way, machines can build themselves if you can achieve that. So it was a Turing machine with a tape copier and a method of inserting itself into the new machine. I have to tell you that this is in a book which was published in 1930, in the late 30s. So in the late 30s, and it precedes, of course, the whole of Watson Crick. The amazing thing is that neither Crick nor Watson had ever heard of von Neumann, and of course von Neumann hadn't heard of them either. Okay, so when the DNA model appeared, which, as you know, were the complementary base pairing, you see, many people had thought of this before, and they tried to copy it. They tried tape copiers, which is like you can just record. You can have a punch that reads one tape and punches a second tape, like in the old days when things were run on tapes, real tapes. They were punched with a set of holes, and that you can do. But they realized that to have a copier, it is far easier to have a copier if there is what is called complementary copying, so that you have a positive and a negative together, all right? Then if you separate them, it's called complementary replication. You can put the positive with the old negative and the new negative with the old positive. It's much easier to do that than making a standard copy. That was written in a paper, again, before the DNA, 1949. It was written there by Pauling and Delbruck, who promptly forgot about it and did nothing more about it. In fact, although Delbruck, so this was just a toy idea, they didn't see the significance. But the important thing about DNA is that it's self-complementary. Okay? And the base pairs is the key thing. It's really the interaction between the base pairs, guanine pairing with cytosine and adenine with thymine, that determines the complementary nature. And even though Delbruck himself was an author of the paper with Pauling. Both he and Pauling forgot about this. In fact, Delbruck criticized the model of DNA because he said he preferred to have one identical strand and assemble another identical. But they couldn't see how you could specify that because they thought it would be proteins that would do it. Well, as you know, Watson and Crick published their important paper in May in 1953. Now, I came to Oxford in 1952 to do a PhD with the only person I thought that would be good was not a biologist, he was a chemist. Okay, I came really to try to understand the chemistry of the genetic material. We didn't know much about it. I had a friend called Jack Dunnett, who was a crystallographer in Cambridge. It was a crystallographer who was working with another famous crystallographer called Dorothy Hodgkin. And he was working with her. He had a degree from Glasgow, but he spent 3 years in Caltech before, so he knew about the Caltech people that worked with Pauling. He was a student with Pauling at the time. And there was another person called Orgel who was in Cambridge and who came from Cambridge but had a fellowship in Oxford at the time. And we all were members because we were not from the recognized schools, and we all had sort of 10th rate colleges and so on. So we were allowed to go to a place called Halifax House, which was terrific. It was a place where people who didn't have, functional connections with colleges could actually go and eat there at lunchtime. You have to remember, this was at a time when there was still food rationing in England, and so I gave my entire ration to my landlady, who I was living with, so she could give me breakfast in the morning. So we were allowed 2 eggs a week and so on. It was all rationed. You could buy unrationed food, but it was so expensive. It was, like vintage champagne. That is because nearly all the food from in England was sent to Germany, to Berlin, which was, at that time, there was all more nonsense in Germany. So it's with Jack that I learned, and I went one day, just before the basic paper, the Nature paper appeared, I went with Jack Dunnett to see the model. And I have to tell you that it was just a moment of absolute enlightenment, that's all I can say. I had seen the light. And I spent the whole of the next day, the whole of that afternoon, before we went back, talking to Jim Watson, who since that time has remained a colleague and friend for all time. And I resolved to work on this subject, and of course, wasn't it much I could do at the time, and so I started to think, as indeed we began to talk then, about the genetic code. You see, so that linked up. So what did this code, what did the sequence mean in terms of making, which we knew, we didn't know how, but the sequence describes the genes describe the whole of the properties of the proteins of the cell. So how did you go from this one-dimensional sequence back to the sequence of the amino acids? Because you must realize that in 1952, Fred Sanger got the Nobel Prize for sequencing insulin, which is a protein. It's a small protein. It's got 2 chains. And so we knew that there was an important in doing this. So the whole of gene coding came out of thinking, out of this model. So, of course, we decided that there was one thing we could do, because Benza, who came to work much later and who I met in America, had described the fine structure of the gene. It was a great experiment in which he showed the gene was divided into many, many sites. There just wasn't 2. Most people thought of alleles as 2 big configurations. It was a very woolly idea. But Benza said it's a one-dimensional message. That's how it connects with the DNA. And if you change the bases, you change the reading. Okay. Then, so I started to collect amino acid sequences, which I did, and there were very few. Then, then Gamow had the idea. So Gamow was a great physicist, very imaginative guy, but had no knowledge of, of biochemistry or anything, conceived of what he called the overlapping triplet code. Okay, and this was that the code he'd learned that the spacing between adjacent amino acids, which is about 3.3 angstrom units, is about the same as the spacing, sorry, it's 3.5, about the spacing between the bases, okay? And I used to ask students, when they thought about DNA, I used to ask them questions like, what lies between the bases? And I got all strange answers, because they imagined them to be these plates of metal, thin plates with a space, you see. And I used to tell them to go and read about anthracene, okay? because there's nothing that lies between the bases. The bases are that thick, okay, because that's the way carbon atoms aren't ultra thin. So there's nothing. I mean, of course, I got all kinds of things like vacuum, air, water, you know, but it's wrong. You see, you have to realize that the pictures of these that you see, like the staircase, is wrong. It's a solid object in which the bases pack on top of each other, you see? And that's a false picture. So Gamow, when he drew this out, suddenly realized that you could have diamonds, you see, which involved three bases. The diamond involved one base pair, the one base on this strand, and the preceding one on that strand. And then you pack these diamonds behind each other, and that was his. And he thought these diamonds made cavities that the amino acids could fit into. But it turns out that was a nonsense idea. He also didn't have any polarity. You could read his diamonds any way you liked. So, but he was the first to make a thing, and he showed how you could take the 64 diamonds, okay, and degenerate them to be 20, of which some diamonds were equivalent, okay? And those were the overlapping triplet codes. And so as we proceeded with all of this, there was a lot of theoretical coding schemes. For example, Francis Crick proved that the code proposed by Gamow couldn't explain insulin. It just didn't have the right things. And so, but there were all different, or hundreds of ways of degenerating the triplets and making them equivalent. You could have as many as 12. However, I realized that if you want to make two peptides together, make two amino acids, a dipeptide, you would use 4 bases because it would be 2 overlapping triplets. That means you could only have, using 2 diamonds, you could only have 256 dipeptides. There would be a constraint on this. And so I realized very early that the way to check the overlapping clone is to show you can't have enough of the diamonds to do this. And I went around collecting all kinds, I mean, even tiny fragments of information from the hydrolysis of albumin would give me a linkage. And I collected all of this because there was nothing that was, there were people engaged in trying to develop protein sequencing, but there was nothing. However, I managed to give a Poisson proof to say it would be entirely improbable that you would have this, which I told Gamow, who promptly stole it, printed it in a review, and said, this was first told to us. So that, but you see, Gamow was so anxious to try and solve the code by theoretical means. And in fact, we proved eventually there was enough protein sequence information for me to write a paper called the impossibility paper, that it is all Gamow codes could be eliminated. All overlapping triplet codes could be eliminated, which then put us to see that exactly how did you read the code. It was Francis who created the adapter hypothesis. What he assumed, very little evidence for this, is that there were short RNAs, short nucleic acids, which would then line up with whatever it is, and which would, of course, they were later discovered. It's tRNA, and there's a whole thing flows from that. But the adapter hypothesis, which he gave in a talk in London in 1958, was poo-pooed by all the biochemists. They said if there were 20 RNAs and 20 enzymes to couple an amino acid to each adapter, we biochemists should have already have discovered them. But since we didn't discover them, they don't exist. So, however, it didn't take too long to show that they were wrong. And so these enzymes were discovered. The whole of tRNA was discovered. And of course, the question is, how did that all work? So, of course, for a long time, it had been thought that the DNA directly coded. And Francis Crick and a man called John Griffith and Leslie Orgel wrote a paper called the Commonless Code. They then showed that there was a neat way to take 64 triplets and turn them so that only 20 of them were what is called sense, and the other 44 were nonsense. And they thought this was so elegant, but as it turned out, it was completely wrong. And so you have to beware in biology that sometimes the magic numbers don't work. They work in many cases, magic numbers of symmetry and so on, but sometimes they don't. And that is one of the cases. So the question then arose, what does this read? And so you all know the story that we discovered with phage-infected cells, there was a messenger RNA that was worked on independently by Jim Watson, but we worked on phage, in which it was but the important thing was exactly how that dawned on us. We were wrong to think, and we had to pose this RNA copy of the DNA, which the DNA can make, so that the tape was actually an RNA copy of one strand of the DNA. Only one strand carries the information for protein. It was a tricky experiment to do because everybody was in doubt about everything, so it couldn't be just shown. But we recognized that by using a technique which allows you to label old RNA, the same technique Messelson used to determine pre-existing strands of DNA by measuring their density when they were substituted by heavy isotopes. So what we tried to do, and did this together with Jacob in Caltech, was to try and show the new RNA went into old ribosomes. You didn't make any new ribosomes. And not a single experiment worked. Everything broke up until it dawned, right on the last day, it dawned on me that we were taking ribosome, which was supposed to be stable, and putting them in 8 molar cesium chloride, so that the value of magnesium was just not enough, and they just degraded during the experiment. The centrifuge had to run for about a day and a half in order to come to equilibrium to make the gradient. So we rushed back to the lab to do the experiment again, and we managed to do it. And so we showed. Then, of course, there were lots more experiments. We had to show new protein is also on the ribosomes, and that there wasn't even a minority of new ribosomes. So that then said, well, there it is. You make a copy of the gene in RNA, and the protein is synthesized in ribosomes with these adapters. And so you might as well have said that on that day, whenever it was, that was the end of it. That's all done. And now we can go on and solve other questions. But of course, as you all know, molecular biology just went on and on and on, and there were more things to get to this. But the basic things of information transfer, which are the fundamental thing of which von Neumann said you had to have, of which is you needed the logical equivalent of a Turing machine with a tape that you could copy and transfer, all right? That is the logical, and that is exactly what makes biology different from physics and from chemistry. Of course, it obeys all the laws, thermodynamics and all of this. That's all obeyed. But what makes it different is this notion of information, which is the unique thing that biological systems introduced into this, and of course introduced it to the science. So if by 1961, still is quite recent, everything was mapped out, we had to then, that is the basic, and you know the rest of the history. The code was solved, tRNA, there was a lot of work. But I decided now must go get on with the next job, okay, which is, you know, bacteria are all okay. And the one concept that you should do and really hold in your head, which I think is terribly important for thinking about higher organisms, is this concept of a cubic micron. Okay? Because what I want you to really understand is that in the space of a micron cubed, diffusion is so fast, you don't need any directions, right? If, of course, the cell, a bacterial cell, is exactly that size. And there are many compartments, so that they rely simply on random motion. Okay? You don't have to, as many people thought, have a machine that, you know, I hand this to you and you have to hand the next thing. Collisions are so fast that even if the chances are that they can be, you can calculate them. So that this then makes the whole of molecular interaction different at those dimensions, okay? Because you don't have to take an enzyme substrate, and direct it to the active site. Because, of course, most of the time, it's banging against the wrong protein. Most of the time, it hits the right protein, it hits it in the wrong place. But even the tiny success of it will give you the correct rates. So diffusion is a potent force. And that means, and if you go through compartments in a cell, you will see that the moment they have enclosed space, like a synaptic compartment, it's about a micron. All of those things now enable you to use molecular collision. So I remember explaining this to another famous mathematician called Gödel at Princeton at a meeting. And you know what he said to me? He looked at me. Gödel is a very famous mathematician. He looked at me and he said, "That's the end of vitalism." End of vitalism. You have to have no forces. Just everything works by confined space. Don't have to direct the things. And that means then that we have, now we can understand all the thermodynamics. And as you all know, this entire business of intermediary metabolism and the whole thing, of course, a lot of metabolism takes place inside mitochondria, which are just ancient bacteria through which can do all these things because it's a confined volume. And you can rely on diffusion to do this. So I think the scene is set for thinking now, at least at the elementary level, collisions, molecular recognition, those are the important things. And if we now go and ask ourselves, what do you need to make a bacterium? How many genes do you need to make a bacterium? Well, they're sort of degenerate bacteria, which have been done with possibly 800 genes. But they need lots of things in the medium that have to be, they mycoplasma. They are really partial bacteria. But the total free-living bacteria, if you go and study the existing ones, about 1,800 genes are required to make the prime photosynthetic organism is a bacterium that lives in the oceans. It has 1,800 genes. And many of the primitive archaeobacteria are 1,800. That is the basic. They can make everything they need from elementary compounds. Right? They don't need any help. Mycoplasma need amino acids, but these make everything. So 1,800 genes, and so it's good because now we would know how to build the first free-living organism. And they are extremely old and ancient. Okay, and some of them, they can live in all sorts of things right up to boiling water. There are organisms that live where the temperature is 130 degrees centigrade. They don't fall apart, and they can work. So from that, all the way down to bacteria that live almost at freezing point, we have a scale of functions. Now, if you look at your kind of bacteria that you read about in microbiology, you will find they have something of the order between 3,000 and 4,000 genes. E. coli is 4,200. Bacillus subtilis, another one, is also about the same number. So what is this extra 2,000 genes doing? What's it for? And it's quite interesting to realize that what has happened is that bacteria have got a huge number of regulatory genes, of genes that switch off things when they no longer are available. You don't waste energy and protein on making them. You turn them off. And that is a later evolution. So strains like E. coli can live on minimal medium. Just give it a bit of sugar and some ammonia, and they can live completely. But of course, they have a way of when they get something free, then they can actually turn on the things to use those free things, because it saves them making their own energy. All right? And we can do lots of experiments on this. And I just give you one example. All right? When they built the first deuterium water, heavy water, moderated, because a deuterium atom contains 2 neutrons, 1 proton. When they built that, it's a stable isotope, but it allows, it actually will moderate any neutron reaction, which is what goes on in a pile when they're trying to do that. To that, they didn't bother to sterilize the water at the Argonne lab, and to their horror, in this water, they suddenly found there were algae growing. The algae were growing on deuterium. And indeed, if you take algae, or even E. coli, you can adapt it to grow on 100% deuterium. So you can substitute all the hydrogen in a bacterium with deuterium. Now, if E. coli has to use the deuterium, it has to break a carbon D-bond, that's tougher. So it slows the reaction. So they grow slowly on this. And there have been a lot of experiments to see, can you adapt E. coli, and indeed any other microorganism, to grow on deuterium such that if you put it back in hydrogen, it would just blow up. See? So the idea was, boy, that's a great way to make a completely safe bacterium, which would not... I have to tell you, those experiments have never worked. It always comes back to hydrogen, but it would be a great idea to make bacteria which can only grow where you want them to, so you can do experiments and not get these loose in the world. But it never worked. And I did a lot of experiments on this to try and get a completely safe bacterium, because no one at a certain time would allow you to do cloning except in a high containment lab, which is really terrible to work in. We used to define high containment as follows, that you see everything going into the lab is sterilized. So then we had even higher containment where you sterilize the operator. You autoclave him before you put him in. And also when he's finished, it comes out, you autoclave him. It was called P6. P4 was the highest grade. It's where you work with really dangerous bacteria and plague. But many of them did escape. And so the whole idea was to get things to work perfectly safely. And that's an episode in my life that we try to get this genetic engineering of making things so that people would not be constrained and could increase the number of experiments. But everybody then had rules, and there were rules. And of course, one of the things they didn't realize, see, Americans make rules. They were all ridiculous because according to them, the danger was the object himself. So I wrote a paper once which said that if you want to work with lion DNA, you need a much higher containment because lions are pretty dangerous, you see, than you do to work with pussycat DNA, you see, which is what they were saying, to work with malaria. You see, whereas we were arguing, when you came to do work with the AIDS virus or one of these, Zika or any, the best thing is cut it into pieces and keep them all separate as clones. You see, that's the safest way to deal with Zika virus. The worst thing you can do is keep them all intact. Okay? And in fact, but it took years before we got people really to think this. So I actually did invent a contained bacterium, and to prove it, I did experiments on what I called was a higher primate, which was myself. I drank the bacteria and saw how they survived through mind testing for days. Yeah, most of them, the survival was only about 10 to the minus 7, which is not bad. It's not bad. But, of course, they said, who advised you to do this experiment? I said I consulted people, and they advised me. They didn't know who I consulted. I sort of consulted people I met in the street. But it's all right. But it helped to release labs from tremendous expense. And still today, because once in Japan they did something, they find it impossible to change. So all genetic experiments in Japan had to be done in high containment because they can't change rules very easily. You see, whereas Americans are always changing rules, simply to suit themselves, you see. But, of course, now it's no longer an issue. We have lots of things. You just asked to be careful. Don't eat while you work with bacteria. You know, eat sandwiches in a lab. So, anyway, I will... so this then takes us to a big expansion of molecular biology and biological sciences throughout the world. But of course, there's some of us who still think they're unsolved problems. And so tomorrow I will talk to you a little, quite a little, a lot, about how we got onto developmental biology, but more importantly, neurobiology. Because once you see this big unit, and of course, there is this thing in your head which is unique. The human brain is a unique biological entity, and we would like to know how that works. So it became, and so one of the things was, you know, when you come in the morning to the lab, I always believe you've got to have an experiment to do. And so we're going to need a new ways of doing experiments. And of course, so we began to ask questions. very early on, in the 60s, what's got the simplest nervous system? But it's got to be a real, it's got to be a real organism and not a toy one, you see. Because people would started to work on chemotaxis as a problem of how bacteria move in gradients that unveiled a lot of interesting mechanisms, but they don't have any brains. Okay, they're not built of parts, you see. And so we needed something which was started as an egg, developed a whole lot of different cells, and did this. And so I think tomorrow I will tell you how we really got to C. elegans from this point, and how we developed the whole thing. And as you know, many people have done lots of work on that, and that is another episode. And I'll talk to you then about this tomorrow to see, you know, what our concepts were at the time and what now seems obvious at the time to do. And then how does this going to be carried forward into higher organisms? And I think and that's, again, now, why did we think the nervous system was important? Because, again, it takes you back to Cambridge, where there were all the people interested in doing things, people like Adrian, Hodgkin, Huxley, all these people were all living in the same small town and interacting this way. And in fact, I think it was those interactions. And I came, when I was still in South Africa, I had to do neuroanatomy. So we had to make models of brains. We had to section brains. We had to learn how to stain them with Golgi and all the other stains. And that was a great tradition there, was to do neuroanatomy, learn how to section brains, learn how to make things with wax sheets. We learned how to reconstruct what's called the bush baby, Galago. It's a little primate that we worked on there. That's a long way from real brains, but anatomy, how the pieces put together. So structure is always important, because structure gives you the clue to information flow. And that's why I think anatomy is a very important area. And that is how the whole business of making maps of nervous systems started. And I'll tell you about that tomorrow. And now if you want any questions answered, I'll try to do things. Okay.
Who is first? Give your name and then
Hello.
I think it works.
Oh, hello. Hello, Dr. Brenner. Thank you so much for the awesome lecture right now. You mentioned that Dr. Pauling and Delbruck came up with the complementary copier theory.
I can't hear you.
Sorry, you mentioned about Pauling and Delbruck coming up with the complementary copier theory.
Yes.
So, as far as I remember, Dr. Pauling was working on the structure of DNA around the same time.
Yeah, he got it wrong.
Yeah,
because he forgot about what he wrote.
It's weird to hear that he had some information which could have given a clue to the structure or to the functioning.
Of course, but you see, that is where even good scientists fail, when they don't believe in what they've really done. Both he and Delbruck forgot they wrote that paper. It was just a theory, you know, it was nice, but in fact, it was the key. So, self-complementarity is the key. Their paper, it's worthwhile going to read it. It's 1949. I don't know, you can get the reference out of one of these things, but it's worthwhile. They were very clear on this, but they did nothing about it. So if you have a theory that's good, you better try not only, if you can't disprove it or prove it, you should do something about it, because otherwise it's a toy. You're just playing games, you see. And that's why the whole of science is full of examples of these, that people who don't even believe what they do themselves.
So as a follow-up, so when the Watson and Crick model came out, and when they proposed the complementary structure, so why were they still why were Delbruck and Pauling still opposing?
Well,
Watson?
You see, Delbruck didn't like it because the strands were wound around each other. So he thought you'd have to unwind tremendous amounts. We now know that you do have to unwind it, but it's not a problem, you see, because we used to have a big thing. They were called don't worry theories. You see, if you say, oh, you have to unwind, the strands are wound around each other, then we say, don't worry, there'll be an enzyme which will unwind it. It's an unwindase, which is exactly what there is, you see. So if you don't see that as an insurmountable difficulty, because nature knows how to solve those problems. So we've had many don't worry theories, you know, that of course everybody says you can't have the strands wound around each other. They've got to be separable, you see, like this. That is why they wanted them parallel, you see. But it's nonsense. It's much harder to solve the unwinding problem than it is to solve this business of how you do parallel information transfer, you see. So the original thought in Delbruck and Pauling is dead right, but they didn't like it. I think Pauling forgot about it in his model of DNA. It was parallel, but effectively that model couldn't exist. There was so much electric repulsive charges between the phosphates because the phosphates were on the inside. You see, that was dead wrong.
Thank you.
My name is Liang Zheng from NTU. So we know that 60 years ago, Dr. Francis Crick, he proposed a principle called central dogma in a conference or symposium. As far as we know, at that time, we don't have enough data or results to support most of the claims in this central dogma, especially the information flow from RNA back into DNA. So my question is, why Dr. Francis Crick was so confident to call his theory as the central dogma? Why not call central theory, or why not call central information flow theory?
Well, you see, he regrets he used the word dogma. But it was a joke, because Francis, like me, didn't believe in God, you see. And of course, the Catholic Church always has dogma. Many churches lay down, this is what you have to believe in. So he made it as a joke. Shouldn't have been called, should have been the central law, if you like. But he made it as a joke. But I'm afraid that everybody opposed it to be a dogma. But he called it the central dogma.
Thank you.
Next question. Who?
Because we now know you can copy DNA from RNA, RNA from DNA. It's okay. It's okay. It's from protein back to nucleic acids. That you can't do. You can't go from a protein sequence back to the code. That's the real thing. Nucleic acids all the same, whether they ribo or deoxy. As we now know, many genetic viruses have RNA as their genetic material. So that was all right. And those are equivalent. It's really not much difference between the information because it obeys that. But to go from protein to nucleic acid has so far been impossible. I mean, there are people who like to claim it, but it's a very, very subtle thing. You can't copy the information directly to DNA from a protein. I can't get the insulin gene back from insulin, except theoretically, you see. But I can't get the material, so that the information flow from nucleic acid to protein is one direction. Okay, and he called that a dogma.
Balázs.
I can ask it from you, Sydney, but let me ask it again. One of the prevailing theories in evolutionary biology is that the RNA world preceded the DNA world. And the reasons are multiple, but including one important thing. RNA, so the ribonucleic acid, can serve enzymatic functions as well.
Yeah.
So what is your read on this?
So I think it's now believed that DNA is rather later than, and that primitive systems existed that were RNA, because the great difficulty was explaining that DNA is very stable, doesn't do anything, right? Whereas RNA can be catalytic. So it's believed that the first enzymes were RNA molecules that could copy themselves and really do things, catalyze reactions. So the RNA world came first. It's a pretty unstable world. So, and you could then argue that once the information was lodged in DNA, you could then make organisms that could exist at 137 degrees centigrade, whereas RNA would be just smashed to pieces. Okay, so that's just, I mean, we have no other evidence. except for that. Okay?
Next one. Come on. You must have been very clear, Sydney. It must all have been very clear, because there seem to be no questions or no arguments.
Yes. There we are. There's another question.
Hello, I'm Abbas from NTU. I just puzzled because you were talking just now about RNA and viruses. What was your knowledge back then about these microorganisms, and how, when they were discovered, if discovered later, like single-stranded RNA viruses or single-stranded DNA viruses, how those were puzzling you? What you thought about them when they came out?
Well, you see, the viruses, the history of virus and virus structure depends on whether you were someone who worked with plants or animals. You see, as you know, RNA bacteriophages were only discovered quite late in the game. So everybody who worked with viruses came from a different background. Plant viruses are lots and lots of RNA viruses. And of course, animal viruses were both. But I think that of course, the virus information tells you that viruses can carry genetic information. So we knew that the genes of many viruses were RNA. But at the time, the big thing of animals and thing was DNA, cells. Okay? So, well, you know, the elephant doesn't have an RNA genome. So it was realized that it was DNA, this rather stable form. But you see, when you see how the different areas developed in history, that a lot of this was unknown at the time. So you mustn't say, oh, I know about these viruses. They were stupid to say this or that. I think it's when you're trying to push a field forward. Okay. You try to get the central account first. And I didn't know that there were viruses, because I never studied viruses. I knew that there were diseases caused by viruses. The common cold was, of course, and flu, you see. But that was only discovered much later than all of this, you see. And ones who realized this and could have a classification of viruses, as you now know, there are single-strand, double-strand, there are sense-strand, the other sense-strand viruses for both DNA and RNA. Okay, so now, of course, we have a lot more information. So, you know, you mustn't blame them for not knowing what took another 100 years.
My question is, how did you react back then when you learned about these things?
Sorry, I can't hear you.
It's more about your reaction when these, I think, might have been puzzling elements to add to the whole...
No, I think, you see, that by the time I got into the subject, viruses were well known. You see, we were doing Watson and Crick worked on virus structure. It was the filamentous tobacco mosaic virus was really, it was known, you see. And so that was okay. But of course we knew that animals had nuclei and the nucleus was full of DNA. That was what we needed to, really explain. So I think that, Crick and Watson also wrote a paper on icosahedral symmetry of viruses. And I think that that's important. The fact that they have equivalent subunits, all of that is very important.
Okay, I think we should call it a day for today.
I
Is that
think
okay?
so, yes. Getting a bit tired.
Right.
Okay.
So,
thank you for coming.
please give.
We'll see you tomorrow. It's in another room.
Right.
Okay.
And we'll have coffee now in the back. And for
can
anybody
have
who wants
coffee.
coffee or tea or anything else, I don't
can
think
have-
coffee, tea or water.
Help yourself.
Yeah.
There at the
Okay,
back.
thank you very much.
Okay.